Okay, so we're going to finish out talking about muscle today and break into the heart. So these last little bits about muscle that we're going to be talking about, we're just going to focus on knowing on a sort of high concept level. I want you to know they exist, but there are going to be more details in the slides than I'm going to expect you to know for these last couple things. Okay, so last time we talked about our different types of muscle fibers from the perspective of how quickly they contract and what way they use to produce ATP. And today we're going to be thinking a little bit more about larger groupings of muscles. We're going to be thinking about control of muscles from the perspective of smooth coordinated movements. Okay. So one thing that we want to be aware of, right, is when muscles move, when they contract, in order to actually move your body, we think of this as the musculoskeletal system, because muscles attach to bone, and specifically they attach to bone when they cross joints, right? We don't usually, I can't think in my mind of any that attach to the same bone in multiple places, right? We need to cross something movable to have contraction be meaningful and useful for us, right? So the way we're going to coordinate activities has to do with different types of receptors in our muscles that tell us things about their position and their amount of stretch so that we can move smoothly using pairs of muscles on opposite sides of joints to kind of stabilize things. So when a muscle moves something, when it contracts, we think of a muscle as having an origin and an insertion. So there's somewhere it starts and attaches, and there's somewhere it ends and attaches, right? The only way that we have for muscles to move the body and to produce tension is by getting smaller, right? We talked about our sarcomeres, and they shorten, right? Or they relax, but they never like push out. That's not actually a thing a muscle does, even though you kind of feel like you're pushing something sometimes when you do exercises, right? The muscles themselves individually can only pull. But we have muscles with antagonistic action, so muscles on opposite sides of a joint that allow us to move a joint, both towards us and away from us, so we can move in flexion or extension at any point. And by bracing ourselves using two muscles or more on opposite sides of a joint, that's how we can kind of stiffen and be able to push, right? But any individual muscle is only contracting. So in order to coordinate activity, your muscles need to know things. In particular, we're going to want to know how stretched out a muscle is and things about its relative position. So we're going to have two things that we want to be aware of inside our muscles that give us some sort of sensory abilities. So we're thinking about our muscle spindles first. Okay. So here we see a muscle spindle inside of our muscle. So this is different than our contractile units, different than our sarcomeres. Right? This is for sensing things, right? So here we can see that we have some intrafusal fibers and some extrafusal fibers, okay, forming our muscle spindle as a whole, right? So within these, our intrafusal fibers can themselves contract a bit, and they adjust the sensitivity of a muscle to stretch, and they are innervated by a specific type of motor unit called a gamma motor unit. Our extrafusal fibers are part of the muscle itself. So those are the contractile cells of the muscle. So this is where you would find the sarcomeres. Okay, so we have these muscle spindles like embedded in our larger muscle. Okay. So that muscle spindle in here is detecting changes in muscle length. So it does this because we're going to see that our different types of sensory endings are going to look kind of like springs, and we'll see how they change their signaling depending on whether the muscle is kind of like bunched up or stretched out. Okay, So we can change their action by changing the actions of these pieces inside of the muscle spindle. So here we see some different states, and here we see relative signaling on the bottom. Right, so here we can see the state of this muscle spindle in its stretched out state, a kind of relaxed, loose state versus a contracted state. So these are all things that you want to be able to sense about your own muscle, right? This is going to help you get to some things about positioning, right? So you can see that as we have this more contracted state, we can see this muscle spindle looking a little more bunched up, a little floppier here, and we can see that these fibers are now getting closer together. And this is changing how rapidly action potentials are kind of going back to the brain, right? So that's how your brain is going to get a signal telling you about the state of an individual muscle. So here again is another comparison, focusing in on the type of gamma or alpha and gamma activity. That's specificity we don't need for our purposes here. The other type of receptor we have, so in addition to sensing the state of like the muscle belly, right? We also have sensation for our tendons, right? So here we have our Golgi tendon organs, right? So rather than being embedded in the belly of the muscle, we're now looking at this tendon organ embedded near our origins or insertions, usually, of the muscle, okay? So what we're activating here is when we feel tendon stretch, right? So the stretch on a tendon activates our Golgi tendon organ, right? So this is going to tell us things like, are we pulling right now? Okay? Now, this is involved in reflex inhibition. So this is involved in reflexes. When we feel stretch in tendons suddenly, we don't love that. And this can protect against overactivity of the muscle. We can tear tendons. We don't want to do that either. We want to have smooth movements, and we want to keep our tendons at a point of tension that they can kind of tolerate. Okay. So in an example here, all right, we're going to think about what would happen if a neurologist is testing reflexes of patient and taps a patellar tendon. Okay. So we have this stretching our quadriceps muscle, leading to a knee-jerk reflex. And we want to think in this case, right, instead of thinking about the tendon organ, we were just thinking about, we want to think about what would be happening with the muscle spindle here. Okay. So in this case, what the muscle spindle is sensing, right, is it's sensing that as the tendon that you tap on gets stretched out, so does the muscle, right? And we have a reflexive reaction in that muscle part to adjust for this by immediately kind of snapping back. So this is part of how reflexes work together. I won't be asking you to do any details on this in terms of exams. But if you want to dive in deeper to this, it really is very interesting. So I recommend you check the section on your book and kind of play around with it yourself as you're thinking about some of the activities you've done, hopefully in the physiology lab, recording muscle activity, and think a bit more deeply about these different types of receptors that help this kind of all work together. So the other thing we want to do as we're finishing out little extra facts about our muscle, we want to be aware that thus far, we've been talking only about one type of muscle. So we've been talking about skeletal muscle, right? So we've been talking about muscle that's under voluntary control. We're thinking about muscle that's striated, so has stripes on it if you look at it, right? But we have two other types of muscle as well, right? So we have two types of muscle that are under involuntary control, so you don't think about it, and have some different arrangements and physical properties when we look at how they're connected to other muscle cells and how they're connected to signaling pathways. So we're going to see smooth muscle and cardiac muscle as two other options for types of muscles, so other types of contractile cells. So we'll see smooth muscle a fair bit when we get to A&P2, because this is a type of muscle that you're going to see a lot in internal organs generally. And in particular, we're going to spend a lot of time thinking about smooth muscle wrapping around blood vessels, forming kind of parts of the wall of a blood vessel and letting us adjust blood pressure. And we'll talk about the concept of resistance, which is another thing that we'll be adjusting using smooth muscle in blood vessel walls. So smooth muscle is this one in the middle. It looks a little different than our skeletal muscle does, right? So we can see it looks different under a slide, and it looks different if you draw a diagram of it. Okay. So one of the things we're going to notice is that it has a different type of nerve coming to it, right? This arrangement of the nerves and the muscle overall is kind of different. This is because it's under involuntary control. So rather than being under the control of the somatic part of your nervous system, this is under the control of the autonomic part of your nervous system. So that sympathetic, parasympathetic part that we've talked about before. The shape of our muscle cells is different in smooth muscle. So instead of having these long fibers with multiple nuclei and these stripes from our sarcomeres, we now have these kind of spindle-shaped or kind of teardrop-shaped cells, okay? And they're a lot smaller. So they're like a tenth the size of a skeletal muscle. They do still have some of the same components. So we would still expect to see actin and myosin, those contractile proteins, but they're actually not even arranged in sarcomeres. So they're arranged in a different shape. So when we think about how these muscles shrink and expand, it would look completely different. I think we have a picture here. Yeah, great. So here we see this kind of lattice work situation, right? So this is what we have in a smooth muscle instead of a sarcomere. Okay. So here we see a new way that we have our thin filaments and thick filaments overlapping, if we were to zoom in here. And we can see these crossing points called dense bodies. Right? So this is a different arrangement. If we were to look in at our myosin, we would see that instead of having a bare area in the middle, our myosin is going to have heads along the whole length of the myosin. And in general, we're going to have like a longer range of contraction. So instead of contracting in these like little sarcomere subunits, we can see that we contract this whole cell kind of overall in this lattice work thing. So we still have some sliding going on. We're still going to have this situation where neither the actin or the myosin itself is actually shrinking as a protein, but the shapes that we can form are going to look different in a smooth muscle than in skeletal muscle. So our innervation, our nerves going here, are either going to be sympathetic nerves or parasympathetic nerves. So our two branches of the autonomic part of our nervous system. Unlike skeletal muscle, where we are only exciting skeletal muscle, right? So all of our signals to a skeletal muscle, essentially we're creating little end plate potentials and action potentials. We were really just sending a signal to contract a skeletal muscle. If we're thinking about a smooth muscle, we can tell it to get excited or we can inhibit it. So we can kind of control both more contraction and control the relaxation side a little bit. What exactly we do to our smooth muscle is going to depend on the receptor types that we see here in our muscle fibers. And we'll talk about those receptors later when we talk about the specific places that we see smooth muscle, especially when we're going through the vasculature later on. When we think about how the signal travels, right, so it's coming down this autonomic nerve, we're going to see neurotransmitter being released from the structure called a varicosity instead of those axon terminals or terminal boutons that we've seen before. And we're often going to see this kind of diffuse neurotransmitter spread. And I'll show you what I mean by that in a second. But it's going to be less like of a pinpoint action on a specific spot and more kind of a broad signal that gets like a little bit sprayed out. Within our smooth muscle, we'll actually have two types. So there'll be both single and multi-unit smooth muscle. So the single unit smooth muscle is more common. So what we're looking at here is that arrangement. So we can see that we have a big long autonomic nerve over the top. Here are the varicosities, right? So these swellings are where we have neurotransmitter. You can see that we have multiple of them in a line, all in the same neuron, right? So instead of just having an endpoint, right, we've got multiple spots where we can release neurotransmitter. And we can see that these muscle fibers in this single unit smooth muscle are connected to each other by gap junctions. So gap junction, remember we mentioned in our neurons unit that there are electrical synapses in addition to the chemical synapses that we focused on. Those were formed by gap junctions, so basically holes or doors from one cell into another. And so this is that same type of connection, that same type of kind of hole or passageway from one cell to another, where we let cytoplasm flow straight to the cytoplasm of another cell. Right, so we see gap junctions here between the fibers, between the cells of our single unit smooth muscle. So this is the most common type of smooth muscle we see. You'll see this in your intestines, you'll see this in blood vessels, you'll see this in the respiratory tract, so when we're breathing. Okay, so here we see those gap junctions. Another interesting thing about single unit smooth muscle, but also we'll see this about cardiac muscle as well, is that these muscle cells don't actually always need a nerve to tell them to contract, right? So they don't always need nervous input in order to contract and relax. Right? So they have what we call pacemaker cells in them. So pacemaker cell is one that just has this kind of natural cycle where we see spontaneous depolarizations, right? So remember, depolarization, the change to the membrane potential, right? So we become a bit more positive, right? And that's how we move towards contraction in any of our muscles, right? Now in our single unit smooth muscle and in our cardiac muscle, we'll see that some of the cells just start doing this without any signal from a neurotransmitter or from a nerve. So this means that this muscle can kind of contract separately from getting any specific nervous impulse. So that's not something you would see a skeletal muscle doing. You're not going to see your biceps start to just kind of like beat, but you will see that in a heart cell on its own in a dish without a nerve, right? You could see that in smooth muscle as well. So in this single unit smooth muscle, we only have innervation actually to a couple of the cells. So notice that this nerve is just lying on top of this patch, right? Rather than going directly to each of these cells. And that's on purpose in this depiction. That's accurate, that's true. So the reason for this, right, is that once we get the signal going to those cells, it can spread through those gap junctions to everybody else in this patch, right? So this innervation is going to trigger the whole unit, this whole single unit smooth muscle, even though it's only touching a couple of these cells. Single unit smooth muscle also has what we call tone to it. When you say someone is toned, right, you say, you know, like you can see a little bit of muscle. What we mean when we talk about smooth muscle having tone, we mean it's like a little bit contracted kind of all the time. Like its baseline is a little bit tense, right? So this is why it matters that we can excite or inhibit our smooth muscle when we send a signal, right? Because we're starting out a little tense, right? So this is why it's helpful to be able to have a signal that says, okay, get less tense, or okay, get more tense, right? That's because we have this baseline tone. We're not relaxed and we're not fully contracted at our baseline state. So we can see both increases and decreases in the tension that we see here. There are also reflexes in our smooth muscle. So when we see smooth muscle get stretched out, we see a reflex that makes it relax, basically. So it bounces back in these reflexes. Yep. Now, we can also see graded contractions in single unit smooth muscle, but these don't have to do with different motor units like we see in skeletal muscle. So in skeletal muscle, we had recruitment of smaller motor units, then medium ones, then larger ones. The way we have graded contractions in smooth muscle is we have different amounts of calcium released. Multi-unit smooth muscle is similar, but the main things you're going to notice are that we have more nerves going through them, and we don't have those gap junctions, which is why we need more nerves, right? So a multi-unit smooth muscle is still located in the respiratory system, but particularly it would be in big airways rather than throughout. Will be in big arteries, and we'll see it in some places inside the eye, okay? So for multi-unit smooth muscle, the difference that this arrangement causes is that these cells are going to react individually, right? So multi-unit is for these multiple cells reacting separately, versus our single unit, all these cells react as one single unit when we send a signal. Okay. So every fiber is going to get its own source of innervation. It's going to be a little more similar to skeletal muscle in that way, and also in the fact that we don't have tone to it, so it doesn't have this kind of like baseline level of tension. And we do see kind of ramping up recruitment because we have to get on more and more cells to get more and more contracted. You don't have a slide about cardiac muscle in your packets, maybe you do, but I just want to mention it to you because we're about to talk about the heart. And you do certainly have this table in your book, right? So what we want to know about our cardiac muscle is that it's going to look similar to each of these other two types of muscle. So like our skeletal muscle, it's going to have sarcomeres. It's regulated in a similar way to our skeletal muscle with this troponin, tropomyosin regulation of those sarcomeres. But like our smooth muscle, and that single unit smooth muscle, it's going to have gap junctions. It's going to have pacemaker cells. So it's going to be able to beat on its own. It's going to be able to contract on its own. And it's going to be innervated by the autonomic nervous system. So those parasympathetics and sympathetics. Right? It can get calcium from multiple sources, and we'll talk about that. And we'll see that its action potentials relative to its tension, they're approximately equal, which is a little different than we see other places. We talked about summation in skeletal muscle, and that's not something that we're going to see in cardiac muscle, which will be good for you because you don't want all your heartbeats merging together into one long heartbeat. That would not be very good for moving your blood around. So we'll be talking about the cardiac cycle and how we get this going later on. Okay. So here we have a table pulling all these things together. So what you'd want to be able to do is recognize the type of muscle you're looking at, right? So striations will be pretty obvious, stripes, which are really just sarcomeres. You'll see in skeletal muscle and cardiac muscle, but not the others. We're not going to worry about actin and myosin presence because, as you notice, it's in all of them. We do want to know that skeletal muscle is the only one under voluntary control. It's also the only one that's hooked up to our somatic nervous system. Everybody else is controlled by sympathetics and parasympathetics in the autonomic nervous system. This means that in skeletal muscle, we have a neuromuscular junction that's very specific to skeletal muscle, but our other types have these varicosities that are part of that autonomic nervous system. We'll talk about hormones later on. We're not going to talk much about where the calcium's coming from or these regulatory proteins in particular, but you can dig into them if you're curious. You do want to know about the gap junctions being present in our single unit smooth muscle and the cardiac muscle, because it's important that they can coordinate their activity. Okay? And we want to know about those pacemakers in particular, because we will be thinking about, again, coordinating activity in our cardiac muscle as we think about the heart, which is what we're going to do now. So we're going to go through some basic features of the cardiac part of the cardiovascular system right now. And I've made an activity for you that'll be due after break, putting together some of our basics of kind of like heart anatomy so that we have a firm foundation for that when we talk next time. So you can do it over break if you want, but it's due a couple days after you get back. So you should have time. So generally, when we're looking at the cardiovascular system, we're going to think about the cardio part, the heart part, which is what this last part of A&P1 is going to be about. We also want to think about the vascular part, which is going to wrap us around into the beginning of A&P2. Okay? But generally, we're thinking about the heart itself. We're thinking about blood vessels coming out of the heart and then traveling through the rest of your body. And we're thinking about the blood that travels in the heart and the blood vessels. Overall, the point of this whole system is to transport things around your body, right? We want to move stuff. We want to move oxygen around because now we know that different cells need different amounts of oxygen depending on what they're doing. But in general, our cells need oxygen to produce ATP. So they have energy to do their various functions. They also need nutrients, so things like glucose, again, to fuel that cellular respiration, that glucose metabolism. We also need to remove waste products, right? For example, once we break down glucose, we watched as we saw like carbon dioxide just kind of peeling off the sides of various parts of that cycle. You have to get that carbon dioxide out of your body. So we're going to see that that's one of the big things that we think of as a waste product that eventually gets to your lungs and you breathe out. But there are other metabolic waste products that we're going to process through our liver and our kidneys. We also need to send signals through the blood, right? So we're going to be thinking about hormones moving through the blood in the endocrine system that we've already talked about. We'd also have immune cells traveling through the cardiovascular system. We're going to think about the clotting proteins that travel through the cardiovascular system. So basically, we've got a lot of stuff in the body that you produce in one place and it needs to go somewhere else. And our cardiovascular system is the highway for getting it there. Okay. So here we're looking at the structure of the heart. So I'm going to have us watch how the heart pumps here, because this is ultimately what we're trying to understand, right? This is why we're listing the parts of the heart.
Oxygen, blue blood, returns to the heart after circulating through your body. The right side of the heart, composed of the right atrium and ventricle, collects and pumps the blood to the lungs through the pulmonary arteries. The lungs refresh the blood with a new supply of oxygen, making it turn red. Oxygen-rich blood, red blood, then enters the left side of the heart, composed of the left atrium and ventricle, and is pumped through the aorta to the body to supply tissues with oxygen. 4 valves within your heart keep your blood moving the right way: the tricuspid, mitral, Pulmonary and aortic valves work like gates on a fence. They open only one way and only when pushed on. Each valve opens and closes once per heartbeat, or about once every second. A beating heart contracts and relaxes. Contraction is called systole, and relaxing is called diastole. During systole, your ventricles contract, forcing blood into the vessels going to your lungs and body, much like ketchup being forced out of a squeeze bottle. The right ventricle contracts a little bit before the left ventricle does. Your ventricles then relax during diastole and are filled with blood coming from the upper chambers, the left and right atria. Then the cycle starts over again. Your heart is nourished by blood too. Blood vessels called coronary arteries extend over the surface of your heart and branch into smaller capillaries. Here you can see just the network of blood vessels that feed your heart with oxygen-rich blood. Your heart also has electrical wiring, which keeps it beating. Electrical impulses begin high in the right atrium and travel through specialized pathways to the ventricles, delivering the signal to pump. The conduction system keeps your heart beating in a coordinated and normal rhythm, which in turn keeps blood circulating. The continuous exchange of oxygen-rich blood with oxygen-poor blood is what keeps you alive.
So the heart has lots of parts to it, and we're going to be running through kind of all of them eventually, right? But what we're fundamentally trying to understand in this unit is the order that blood travels through the heart and these blood vessels and why, right? So we're going to be thinking a lot about pressures in different places, and we're going to be thinking about volumes of blood in different places. So here we have a full kind of anatomical picture of what the heart looks like. So we've taken a heart and sliced it in half to try and see the different chambers. It can look a little messy this way. When we're talking in class, I'm often going to be drawing the heart just like literally a heart, right, with a box inside it, right? And I'm doing that just for simplification purposes, but I'm going to show you what we're kind of linking up when I do that. This is not to scale clearly and not quite to shape either, right? But we're going to see those four chambers, right? We have two atria and two ventricles, and we have a right and a left side of the heart, right? So here on the right side, where we see our deoxygenated blood coming in, the first place we go is our right atrium. So when I draw a little picture like that, this is our right atrium where blood is starting out. We're going to see that then we have a valve that passes us into our right ventricle here. Okay, so that's our next chamber. All right, so down here we have our right ventricle. Okay. And from there, we then send blood out to the lungs. Right? So from this right ventricle, when the heart beats, we send blood through a series of tubes, right? So through a pulmonary trunk, a valve in that blood vessel called the pulmonary semilunar valve, out to the right and left lungs along these pulmonary arteries, right? So from here we go to the lungs, okay? And then we're going to circle back from the lungs, right? So we are going to come, again, every time we enter the heart, when we're thinking from the perspective of blood, we're going to come into an atrium, right? Just like a building has an atrium, right? Like vestibule kind of threshold area, maybe where a waiting room of a doctor's office is, right? That's the atrium, right? So this is the waiting room of your heart, right? So this is the left atrium over here. Okay? So we can see that here-ish on the diagram. So here we see veins coming from our lungs back to the heart. And then we're going to pass through another valve down into our left ventricle, okay, before we go out our biggest artery, the aorta, out of the heart to the rest of the body. So this will be our aorta out to the rest of the body. So we'll get to know all these other structures in the heart, the different types of valves, and in A&P2 in lab, you will hold a heart in your hands and see the different structures that you see in the walls of each of these chambers. But when we're starting out, start with the basics and we're going to add stuff on. We'll also be adding on the conduction system later on in this unit. Not very visible, not at all visible, if you just slice a heart in half. This is going to be a pathway of different types of cells with the ability to send electrical signals. But unfortunately, they're not bright yellow like they are in diagrams of the conduction system. Okay. So our heart is our big pump, moving blood through the body, through the different pathways, sending us to organ systems and then coming back. So when we think about vasculature, we think about a lot of branching, and we think about things first getting smaller as you have branches splitting and splitting and splitting. And then we start to have them collecting. Once you get to your smallest blood vessels, the capillaries, they're going to start bunching together and getting bigger again before we get back to the heart. So for our order, coming out of the heart, we're going to have our big arteries. They're going to be really big. Things like the aorta, taking blood away from the heart. You can see an artery in the body. Okay? Our next level of structure, eventually we're going to branch down to the point that we get our arteriole. You can't really see arterioles with the naked eye. You have not been naming any arterioles in lab, but our arterioles will eventually be very important for regulation of things like blood pressure. So these are going to be smaller branching vessels where we're going to be able to essentially alter how tight those blood vessels are. They're going to alter their resistance. Our capillaries are going to be where we have like fluid and nutrients and gases mixing from the blood to different types of tissue. So those are specialized for exchange. And then we start coming back. So one of the things we exchange are gases. So that's where we dumped out oxygen to the tissue. So now our blood is deoxygenated and it's going to come back first through a venule. So I like to look at that L at the end, think little vein, just like I like to look at the L in the arterial and think little artery. All right, so we're going to go from a capillary into a venule, basically a little vein, and then into a larger vein that you could see with the naked eye, eventually getting us back to large arteries coming into the heart, coming into that right atrium. This is a closed loop. You don't have any like big gaping holes where you have blood leaking into your tissue. If you do, that's a problem. So all your blood needs to be circulating through your heart, through your lungs, and through the rest of your body. The blood itself is made of different parts. So one of the parts that we talked about already actually was the plasma. We mentioned that that's one of our types of extracellular fluid way at the beginning of this course. So that takes up a little more than half of your whole blood volume is that plasma, that extracellular fluid. Okay, it's going to have things dissolved in it. So it's going to have ions. It's going to have a specific pH. And we'll talk about how we regulate that in A&P2. We're then going to see the part that has cells, right? So this big part on bottom in red are your red blood cells or your erythrocytes. These are what are going to be like our vehicles for oxygen and carbon dioxide. So a lot of what we think about when we think about blood is about gas exchange. So that pairs us up with those red blood cells. And then this tiny little line here in the middle, kind of lighter, called the buffy coat, is made up of our leukocytes and our platelets. So our leukocytes are white blood cells. So we're thinking immune system there, defending your body against diseases, against pathogens. We'll think about types of white blood cells in A&P2 as we think about types of blood cells more generally. Then we also have platelets in there, which are going to be important in blood clotting. But that's a very small component of your blood. They're really important, these white blood cells and platelets, but they're less than 1% of the volume of your blood. Their blood, all of those pieces, are flowing through this closed loop system in two circuits. So you're thinking two loops, right? So we're going to have a pulmonary circuit, which is the part that flows blood to the lungs. And then we're going to have a systemic circuit that goes through your system, aka the rest of your body. So from going through the chambers of the heart, we already know that that pulmonary circuit is getting blood from the right half of the heart, right? So we have blood vessels going from the heart to the lungs, and then from those lungs back to the heart. So in that pulmonary circuit, our blood vessels going from the heart to the lungs are deoxygenated. We haven't picked up oxygen yet. And fun fact, arteries, the word arteries, are always named for the fact that they go away from the heart. So think arteries A, away A, right? So the blood vessels that go to the lungs from the heart, even though that blood doesn't have any oxygen in it yet, they're going away. So those are arteries. So those are the only arteries carrying deoxygenated blood in your whole body, right? Which means that the blood vessels coming back to the heart is always going to be a vein. Those are your only oxygenated veins in the body, as we have pulmonary veins coming back to the heart. Now, we think of these two circuits as having different types of flow. So we think about this concept of series flow and parallel flow. We're going to take a look at what that means. All right. So when we think about series flow, right, what we're thinking about is that blood has to flow in series, aka like in a specific order, right? So when we say that two things are in series, circuits are in series, right, we're thinking that we have to have step one go out to the lungs, right? And then step two go out to the body. Okay. Then we loop back up to the heart eventually. All right. So that's a series flow. First thing, then second thing. Okay. So we have our blood flowing through first the pulmonary circuit and then the systemic circuit in series. But when we think about the systemic circuit itself, right, if we're thinking about what's happening when blood leaves that left ventricle through the aorta and goes to your gut, goes to your liver, goes to your left knee, right? This is where we start to think about parallel flow, right? So in the systemic circuit, you don't have to go through first the brain and then go to the intestine and then go to your pinky toe, right? We see that blood, because of this branching system, gets to go to all those places in parallel, right? So that's what we're looking at here as this parallel flow, right? So we have arteries that go to the brain and then come back from the brain to the heart. We have arteries that fuel the heart muscle itself, lose their oxygen and come straight back to the heart to get more oxygen. We have blood that goes to your skeletal muscles and then comes right back to the heart. There are a couple places we see series flow in the systemic circuit, but they're really specialized. They're the exceptions rather than the rule. So it is the case that when we're thinking about our gastrointestinal tract, some of that blood has to be processed through the liver, but only the liver, before it comes back to the heart. This is important because you don't want everything that you eat to end up in your blood, right? The liver has some important roles in making sure you're not internalizing toxins and pathogens, and it helps kind of process some of the things that you absorb. But on the whole, we're going everything independently in parallel, right? That's why we have these equal lines going out to our different organisms, or organs, I should say. So each of these would have an artery going through it, branching into its own arterioles, branching into capillary beds before they come back in venules and veins, then to the right side of the heart. Okay. All right. So let's make sure we were all awake. So which way do we have blood flowing to our organs in the systemic circuit? Are we going in series, 1, 2, 3, 4, 5, or are we going in parallel, splitting out the separate organs? Okay. So look. Ooh, okay. So what we want to remember, all right, series is that we have to go through one thing and then another, right? So this is going to be specific to when we're thinking about the fact that we have to go through pulmonary system first, pick up our oxygen, and then through the whole systemic circuit, right? But within the systemic circuit, we're trying to flow to separate organs separately. So that's why we have parallel flow, right? So we're thinking we have a big blood vessel, right? And we're going to have one part going to like our thyroid gland, let's say. We talked about glands, right? And then we're going to have a separate branch of that blood vessel going to our parathyroid gland. And these pathways are in parallel rather than blood having to go through one and then the other. So as we return, we merge these together into the same vein, but these two pathways were separate. So that's what we mean by parallel flow through the systemic circuit. Okay, so you'll take a look at some of that stuff in the activity on heart stuff that you'll be doing after you return from break. Okay. And we'll pick up where we left off.